All right, for this segment of screencast notes, I wanted to go over the thought process underlying Mendel's monohybrid cross and introduce a new concept for you called a Testcross. And so you would hopefully then, after these notes, be able to explain what is a Testcross and what is its purpose, or what would its purpose be. All right, so we left off with this question yesterday: when do homologous chromosomes segregate or separate? And you guys answered in class, and you said meiosis 1, and specifically anaphase 1. So meiosis is specifically anaphase 1. Remember, our genes come in pairs and they can only donate one of their two pairs for a given trait. Okay, so what is the outcome if genotype is homozygous? Well, you obviously can only have the phenotype that's represented. So only one allele gets passed on, but because they're both the same, the phenotype will be identical. What about if the genotype for a trait is heterozygous? That means two different alleles. In this case, both alleles have a 50-50 shot of going into one particular gamete or the other. So this diagram just is showing us anaphase 1, where the homologous pairs are separating, the two colors representing the two parental chromosomes that are in our cells. And so they're also showing you the random assortment of those different parent genomes going one way or the other. So not all the red are going to one side and not all the blue going to the other. And you also hopefully notice that prior to this anaphase 1, the crossing over has happened. And so they have swapped a little of their gene information. All right, so the mathematical model we use to understand these probabilities, you know, of course, as the little window-like thing known as a Punnett square. You showed, most of you understood how to use those. And so then what do we call the measure of chance in a particular outcome? Talk about probabilities. Probability. And what can we use as a conceptual model to show trait possibilities? Those squares we know as Punnett squares. So nothing brand new here. So they're showing the parent genotypes on the outside and the offspring possible genotypes on the inside, and then you interpret the genotypes as phenotypes based on the dominance or recessiveness of the alleles. So here's an example. In this case, you have two parents that are heterozygous for this trait, whatever trait A is. And the capital A is in 3 of the boxes, so 3 out of 4, 75%, you have the dominant allele expressed, or the phenotype. And 25% of the time, this Punnett square shows you the recessive phenotype would show itself. This is a diagram from your book, and not a bad one, just to kind of have an overview concept of what's happening in relationship to meiosis and gene pairs separating. It just kind of visually shows you you're following gene P on the chromosome from these two parents that are going to form gametes, and then the offspring in this case, is getting one version or one allele from one parent, the other opposing allele from another. So they're heterozygous. And they kind of step you through the Punnett square for that process. All right, so how can a genotype of an organism be determined? If we have a purple flower and the purple allele is dominant, the question is, how do we know what the genotype of that organism is? There's two possibilities. It's homozygous dominant or heterozygous. We don't know, so we can do something to try to find that out. And that's where that new concept I mentioned is coming into play, called running a test cross. So a test cross is a method to determine whether or not a trait is in an organism is a heterozygous genotype or homozygous for a dominant version of that trait. So the way that you test that is crossing this unknown genotype with one that you do know. The only ones you know for sure are the recessives. So you take one that's recessive, little p, and cross it. And depending on the outcome of their offspring, when they've been crossed, that will determine what the genotype of the original parent was that you were wondering about. So if the unknown purple flower was big P, what would the offspring look like? Well, it would always get a capital P from that first parent, and so they would all end up with a capital P, which means they would all be purple. So you'd have 100% offspring showing the dominant allele. If, on the other hand, the parent was heterozygous, what would the offspring look like? Well, you would have a probability of 50% of them being the dominant allele, dominant purple, and 50% of the time they would show the recessive. So what do we call test crosses looking at one trait at a time? That's what Mendel was doing. If you're only looking at one trait at a time, it's called a monohybrid cross. So that's checking for dominance relationships between two alleles at a single spot on a chromosome or a single locus. So Mendel started his crosses with true breeding individuals to then develop his monohybrid. So the P generation, then the F1 generation, or the hybrids, and then when they were crossed, then you had the F2 generation. What abbreviations were used to refer to the parents and offspring of the test cross? I just alluded to that, and you would have read about that in the chapter. But P stands for parents, F stands for filial, and that refers to their offspring. And so F1 would be the first generation of offspring, and F2 would be the second generation of offspring. So when you see the P, the F1, and the F2, that's what they're referring to, and they come in that order. So when you cross the plants that drew purebred for white, purebred for white and purple, and all of the offspring, F1, were phenotype was purple, but they were hybrids, they were heterozygous. And we crossed two F1 plants that were both hybrids. Then he got 3/4 of the F2 plants having purple flowers and 1/4 having the white flowers. That's his famous 3-to-1 ratio, and what led him to conclude that principle of dominance, but also led him to determine the principle of segregation. So this is the same diagram, I think, in your book, just outlining the monohybrid cross from the F1 offspring that were both hybrids, big P, little p, and then you see the dominant phenotype 3 out of 4 times, and 25% of the time you get the recessive phenotype showing. These were some other traits that Mendel looked at on pea plants. Just to understand that his work was very detailed, and we often just gloss over 1 or 2 traits that he was exploring, but he explored several. And so ultimately, what did his monohybrid cross tell us? Well, I have mentioned it, but I'll mention it again. So he came up with the phenotype ratio 3:1 in the F2 offspring for his monohybrid cross. And so that's what we consistently see if we use a Punnett square to work out those probabilities. So our use of Punnett squares reinforces the data that he got. He never used Punnett squares. But this is the basis for Mendel's law of segregation, which predicted the following. Mind you, he didn't understand what meiosis was. It hadn't been discovered yet. But he predicted that a process like it must happen in the formation of gametes. So what he said was, diploid cells have pairs of genes on pairs of homologous chromosomes. And the 2 genes of each pair separate during meiosis and end up in different gametes. The fact that some of the alleles disappeared in the F1 generation, that's where the principle of dominance came into play. So what's Mendel's third law or principle? You hopefully know it as the law of independent assortment, and we will save that for another set of notes.